All right, good afternoon, everybody. We are here in a Google Meet to get you pretty much wrapped up with your biochemistry unit. You have been in the biochemistry unit for a really long time. I don't know if you've noticed, it's been like 6 weeks that we've been doing, okay, maybe not that long, I don't know, I haven't looked, but we've been in the biochem unit for a long time. I am hoping to be able to just walk you through your study guide today. And what I've done, and I'm gonna maybe share my screen here really quickly. What I've done is pulled up all of our notes. Is that Ayub? Hi, Ayub. I see you there. Give me just a second. Oh, nice wave. By the way, Eid Mubarak. I don't know if I said that right. To some of our students who have been fasting for the last month, who got to have a festival or celebrate last weekend. So congratulations on that. I'm very happy for all of you. So what I wanted to show you, I hope you guys can see my screen right now. But I opened up, I'm going to click through. Yeah, you guys can see. So I opened up the 5 different sets of notes we've taken in this unit. So we started out just with a junior high review of atoms and protons and neutrons and electrons, that kind of thing. Then we talked about carbohydrates. We talked about lipids and nucleic acids. We talked about proteins, and then we talked about enzymes. In the course of this unit, we also did the biochemistry lab. We did the liver lab. We built, okay, ideally we would have used models and built some of the molecules, but you guys went to a website and put together some atoms to build some molecules. It wasn't as good as I would have liked it to be, but anyway. And so I'm just gonna walk you through your study guide today, and we'll look at the notes and we'll answer the questions as we go. And so you probably want to have something to write with. Are you guys using styluses? I hope that you're using a stylus. I have a really nice pointy stylus, and that makes it so much easier for me to write my answers. Okay, the other thing that I was going to say just really quickly, just for announcements, to remind you, and they've sent this information out to families, but I know that some of you guys are not reading those announcements because they get so long. I totally get it. So number 1, we are done with new curriculum in all of your classes this Friday. So you have 4 new days of school left of the school year, and then you're done. The last week, so next week, is just for makeups. It's just for turning in late work. It's just for if you're bombing out in a class and you need help, that's what next week is for. It's also for graduation. It's also for turning in materials. So you guys have my textbooks. They need to be brought back to the high school. You know, whatever, if you need to pick up in something that you left in a locker, that needs to be picked up. You can drop off if you're renting a musical instrument, whatever. There's all kinds of things that are happening next week. And you need to look at the emails because they're telling you that kind of information. I think sophomores and juniors, one of you could correct me if you know this for sure. I think sophomores and juniors are supposed to drop off and pick up things on Tuesday and Wednesday of next week. So that's that makeup week. And you can also pick up your yearbooks then. So it's going to be a drive up at the high school. They're going to have paras and that kind of thing that are outside. And you drop things off or pick things up as need be. Okay, so moving on, we're going to go through this review today. And then on Thursday, I'm going to give you a biochemistry test, but it will be 2 important things: formative, not summative, and it will be open note. If you are somebody who really needed a summative, like you've been counting on the summative points to bring you from a B+ to an A-, or you've been counting on the summative points to get you up somewhere, talk to me. I have a summative available. And the way I would work that, we could do that the following week, and I would just do a Google Meet with you while you were taking your summative. And we can make that work, but just email me if you're interested in getting some summative points. We'll make that work. Okay, I hope that's all the announcements. Oh, I know, I did have one other thing I wanted to just ask. If you did my attendance check-in today, I was just curious if anybody had any good ideas for the names of the Orioles that are now visiting my house. I got to photograph the female today. She was eating an orange, and I got some really good pictures. So maybe if you have a minute, you could type into the chat room what your ideas were so other people could see what your ideas were. If you don't have a good idea and you don't want to do that, that's totally fine. But I think it would be funny just to see if you guys came up with some funny names or good names, whatever, for the 2 Orioles that have been visiting me. All right, let's go ahead and dive in. I'm not sure the best way to do this. I think, so I have, some of you know I have this program that's called Squid, and I can write directly on my screen. It's kind of like a fancy, let me do a couple things here. It's kind of like a fancy version of Cami. And I really like working with it. I wish you guys all had it. Let's go ahead and work through this. I can write, I'll just write sloppily, and then I'll show you where I'm finding my answers. You should have this assignment pulled open on your screen, and you should be ready to write in the answers, and I will help you go through it really quickly. So I think, give me just a second. I'm going to open it on my other computer so I can go back and forth really easily. Give me just a moment to get that organized. Let's see, that's on Google Drive. I thought I was already, and now I'm changing my mind about how I want to do this. Maybe some of you are still pulling up your assignment, and so that would be okay. And you're welcome to tell me a funny story or say something while you're waiting for me to pull this up. I would ask you about your plans for summer, but I'm guessing that pretty much nobody has plans for summer. Am I right? Like, does anybody have anything going on this summer? Like, is there anything that didn't get ruined by COVID? Feel free to pop in if you have something that's fun that's going on. My daughter is getting her driver's test. I'm super excited. I don't know if any of you guys have had your driver's tests taken away from you, but my daughter is having her 3rd behind the wheel today, right now, actually. She's coming home from that. And then her test was scheduled for the week that we have, well, a couple weeks. It's still a couple weeks away, so it never got canceled. So she actually gets to have her driver's test. I am very sorry for those of you that missed out on your driver's test. All right, I'm going to go ahead and go back then. I think for the moment, I'm going to go back to our notes and we can take a look. So the first thing, question number 1 says atomic number is the number of, and I'm going to pull that right up. So atomic number, oops, come on. There we go. Atomic number is the number of, and then you've got a word bank off on the side. So I'm just showing you on the screen where you can find those answers. So number of protons, atomic mass, right here shows that it's the protons plus the neutrons. So you would want both of those written down. I'm going to go fast, you guys, just to save you time. So just, it's a review and you're writing stuff down. What has a positive charge, a negative charge, and a neutral charge? We're talking, in that case, we're talking about the components, the parts of an atom. And this should be review from junior high. So positive charge on this page, it shows you that the protons are the ones with the plus. The negative charge are the electrons, which we often show with a lowercase e and then a minus sign next to it. And then neutrons are the ones that have the neutral charge. Would somebody holler at me if you feel like I'm going too fast? So just be like, KJ, slow down. And I will slow down, but I'm trying to be respectful of your time. Okay, let's see. Letter F says bonds that occur when atoms share. And what are they sharing? So let's move this. Give me a second. There we go. So when atoms share, an E with a minus sign is an electron. So when atoms share electrons to form a neutral molecule, those are called covalent bonds. You might remember that from junior high. And then the other type of bond is called an ionic bond. And an ionic bond is when the atoms are actually exchanging, that's the wrong word. One is taking electrons and the other is giving the electrons away. So when one atom donates electrons to the other atom, that would be an ionic bond. So that would be for G. Ionic bonds occur when atoms gain or lose electrons. And then H blank are charged atoms that have gained or lost an electron, and that would be an ion. An ion is an atom that has a charge. So for example, Na plus Cl, so sodium plus chlorine, makes NaCl, and the Na loses an electron, so it's actually Na plus. So we don't call it an atom anymore. We call it an ion. Chlorine gains an electron, and so it's Cl minus, and that's an ion. Okay, and then letter I, blank, are more than one atom joined together, and that would be a molecule, and I don't know where I have that here, right here, molecules. Whoops, going slower than I meant. Two atoms and molecules. Molecules are two or more atoms put together. Can anybody shout out to me number 2? Does anybody feel like they know number 2? Blank, 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 and blank make up 96% of living matter. Who feels like they know what 4 elements make up 96% of our bodies and trees and everything that's living? Anybody? Anybody?
Is it nitrogen, hydrogen, oxygen, and carbon?
It sure is. And who was that just said that? I can't see.
Ayub.
Ayub, excellent, good. And you know what, Ayub, you said it in a different order than I usually say it, and there's a reason I say it in a different order. So you said the exact right ones. It's hydrogen, oxygen, nitrogen, and carbon. So everybody should write down H, O, N, and C. And then the next question, question number 3, asks you to draw them with the number of bonds. And I have it pulled up on the screen here. I suppose it's a little bit dark for you guys. But the reason I say it H-O-N-C, I even say the word honk because it sounds kind of dorky, and I like dorky things. H has just one bond, and it's the first letter in the word honk. Oxygen makes 2 bonds, and it's the second letter in the word honk. Nitrogen makes 3 bonds, and it's the third letter in the word honk. And carbon makes 4 bonds, and it's the 4th letter in the word honk. So it helps students remember how many bonds each one needs to make in order to be stable. And then we looked at this, we looked at a couple diagrams on an assignment we had, and we said that, for example, these 2 oxygen atoms are both stable because they each need 2 bonds, and each one of them has 2 lines going in, and so they're stable. But that this one is unstable, and can anybody tell me why this one is unstable? Which atom does not have the right number of bonds, does not have the right number of shared electrons around it? Who can tell me which one is screwed up? Anybody?
Oxygen.
Oxygen is wrong. And was that Tan, maybe?
Oh.
Whose voice was that?
Ayub again.
Ayub, oh, are you just dominating this whole thing? Man, you guys, you can't count on Ayub all the time. Thank you, Ayub, for answering. Appreciate it. Yes, oxygen is the right one. Oxygen has 3 bonds around it. It's got 3 lines coming out of it instead of 2, and it's supposed to have 2. My dog is behind us, and she often does that. So my apologies. Trixie, shush. Let me grab her really quick. Let me grab her really quick so she's not bad. Come here, nice. Oh. I don't know if any of you have dogs at home right now, but Trixie is becoming very dependent. She's so happy that we're home all the time. But now she's barking at everything, and she's just like defending our little territory and whatever. So she's getting to be trouble. All right, we should be done through number 3 right now. I'm going to scooch on. Let's see, we're ready for number 4. This we didn't get to talk as much about as I wanted to, because the presentation that I gave to you guys, the screencast didn't quite have the right screen on it. I screwed that up, so my apologies on that. But this is the right screen that you're supposed to have for this one. So it says the electrons shared by oxygen and the 2 hydrogens in a molecule of water are part of and that's called a polar covalent. So you want to write the word polar covalent bond. This means the electrons are shared unequally. They are not shared in the same way. They are shared unequally. Did somebody just join us? Hello to whoever just joined us. Or did somebody go away? I have no idea. All right, let's see, where are we? So it says the electrons are shared unequally. Oxygen is much more electronegative. So there's the word right here. I'm highlighting it on the screen so you can see it. Oxygen is much more electronegative than hydrogen, so it pulls the electrons with greater force. instead of sharing the electrons evenly. Because of this, oxygen has a slight, and it has electrons near it more often. And since electrons are negatively charged, it has a slight negative charge. So you write negative. And then the hydrogens, because they're losing their electrons, because oxygen is taking those electrons away, the hydrogens have a slight positive charge. And finally, number 5, because water is a polar molecule, the oxygen of one water molecule is attracted to the hydrogens of a different molecule. This attraction creates a weak bond, and that's down at the bottom here. It creates a weak bond called a hydrogen bond. Does anybody have any questions about that water section on our worksheet? Anybody?
I can't find the study guide.
Oh no, it's it should be on Google Classroom.
Yeah, and I've refreshed, but I can't find it.
Are other people having the same problem? Did I screw something up? Because I screw things up on a daily basis. That sounded like Gabby. Was that Gabby?
Yeah, that was me.
Hi, Gabby. I didn't say hi to you yet. Well, let me just ask somebody specifically. Tan, I see you there. Tan, have you been able to get the study guide?
Yeah.
Okay. Gabby, do you have any idea what might be wrong?
No.
I'm pretty sure she's A day, and this is a B day for B day group me.
I have no idea. You guys are just all my students. I don't think about it all. Gabby, that's probably it, honey. You are in the A-day class, and it is a B-day because it's a Tuesday. You are not the first person to have made that mistake. I've already had contact with several other students that were having the same issue. Thanks, Ton, Smarty Pants. So, Gabby, you're supposed to be doing history today.
Okay, bye.
You know what? Do you want to stay here? Because I could just share the document with you. Do you just want to stay and I'll just share it with you?
No, it's okay.
Okay, I will maybe see you tomorrow then.
Bye.
Okay, talk to you later. Bye. Ton, how'd you figure that out? Well played. All right, where were we, everybody? So we're on pH scale right now. So I'm just going to scroll down here really quickly. I don't need you to know a whole lot. You're going to learn about the pH scale in your chemistry class next year. Most of you are going to take chemistry next year. So things that you really need to know, you need to know what's an acid and a base, and that the pH scale runs from 0 to 14, or sometimes they say 1 to 14, but 0 to 14. So it says, number 6, pH is a scale that indicates how acidic, you put an IC on the end of that word, or basic a solution is. The scale runs from 0 to 14. Acids are lower, meaning a lower number. On this picture that's on the screen, acids are actually at the top, but notice the number is smaller at the top. I would not have made the picture that way. That was dumb. On the scale, and then 7, or water, sorry, water is neutral because it has a pH of 7. So pure water, if you look, it's in the green on this diagram, and it has a pH of 7. If you know that much about the pH scale, you're fine. That's all you need to know about it. You'll learn more in chemistry class next year. All right, now we're going to move on. 7, 8, 9, and 10. Compile all 4 of the, or I guess it's 3 of the presentations. So I can't click on any, oh, maybe I can actually, because if we click right to the function, there we go, I can do that. So it says number 7 is carbohydrates, and it's asking you which one is which. So the function of the carbohydrates, those are your sugars and starches, and those are your quick energy. So if you have somebody who's going into diabetic shock, for example, and they have low sugar, you give them something that has sugar in it, like they say orange juice is good for somebody whose sugar is low, and it gives them that quick energy and helps them to start coming out of that diabetic shock. So carbohydrates are quick energy. And then let's see, I can figure this out. Next one, the lipids, which is the fancy word for fat. Their function, it's right here, is long-term energy storage in mammals and animals, not just mammals. So you're storing whatever food. So food is energy. And if you don't burn off that energy, you store it as lipids in your body until later. Proteins, let's see. Nope, that's a different, let's see. What else was that? Oh, nucleic acids are on this set of notes also. So let's scroll to nucleic acids. We learned about nucleic acids. You had an entire unit on nucleic acids. You just don't necessarily remember that. It was called DNA. That was our unit. So what's the function of a nucleic acid? The function is to store our genetic information. So that's down there. And then the last one you can obviously figure out. Proteins is actually the hardest one to come up with a function because proteins are everywhere in your body and they do a bajillion different things. They're involved in structure. So that picture indicates your hair is made of protein, your fingernails are made of protein, but they also do really important jobs in your body. And they are also all of our enzymes. So enzymes speed reactions up in your body so that you have these you have so many chemical reactions happening in your body, and the enzymes are what are allowing it to happen super fast. All right, now the next section is the section that takes the longest, and I'll try to just zip you through it really fast. This is looking at the molecules themselves and seeing if you can recognize the shapes of the molecules or what elements or what atoms are in those molecules. So let's start, we'll look at the carbohydrates specifically. So for example, with the monosaccharides, the prefix mono means one and saccharide means sugar. And so they just have a single ring, and that we call that a ring. It looks like a hexagon or a pentagon. So single ring for monosaccharide, double ring for disaccharide. See if I can show that. Double rings for disaccharide. So here's a disaccharide, here's a disaccharide, and then 3 or more rings for the polysaccharide. So starch is an example of a polysaccharide. So let's see if we can cross off a few of the items, and hopefully you're working while I'm talking. Let's see if we can cross off a few of the items for you right now. So number 11, anything that has one ring. Well, the umbrella, what this entire set of notes is called, is carbohydrates. So for sure, everybody who's got rings, those are going to be the carbohydrates. So for 11, 12, and 13, we would say that those are carbohydrates. So whatever letter that is, B, so you're going to use lots of these answers more than once, and lots of them have more than one answer. So we would say B for all 11, 12, and 13. One ring is a monosaccharide, so that would be J. 2 rings is a disaccharide, and 3 rings is a polysaccharide. So hopefully you're getting those letters written in. You can always come back if you need to on this. And then what are some examples? I think we need examples. Give me a second. Yeah, so an example of a monosaccharide, probably the world's most important monosaccharide, is glucose. That's what plants are making in photosynthesis, and then you're taking that in to make your energy and your ATP. We did not have a disaccharide example, and then for a polysaccharide example, we had starch. So starch is what you get when you eat spaghetti or bread. It takes you a little bit longer to digest it, but it's still your quick energy. All right, let's try number 14, long chains of hydrogens and carbons. Maybe some of you know some of these answers before. I'm going to purposely talk a little bit slowly, and hopefully you can figure some of those out. But here's an example of a long chain of carbons and hydrogens. Fatty acids are types of lipids. So for number 14, we would say, give me a second, lipid is I, and then the saturated and the unsaturated fats would all be examples of long chains of hydrogen. We call them hydrocarbon chains. That's long chains of hydrogen and carbon. All right, number 15 says that there's an R group. Well, the only type of atom that has an R group are the amino acids, and you put a whole bunch of amino acids together and you get a protein. So here's an amino acid. An amino acid is the building block of a protein. Proteins are enormous. They have hundreds or thousands of amino acids put together. And each amino acid has what's called an R group. And the R group means it's variable. Every amino acid is a little bit different. So these are all the amino acids. There's 20 amino acids that humans use, and each one at the bottom has something different, and the bottom represents the R group that's a little bit different. So R group for number 15, you would write down, give me a second, protein, wherever that is, Q. You might have written A, amino acid. That would also be accurate. So you could have either one of those, and I would accept that. All right, number 16, C double bonded to O. There's a whole bunch that have that. So first of all, I see that right here, there's the C double bonded to the O. That's a carboxyl acid group that's part of an amino acid. That's why it's called an amino acid. So carboxyl acid group. So we would say number 16 was amino acid. You could give that an A. It is also present in another one that you recognize the name of. There it is. So the fatty acids, and there's two types of fatty acids. You have your saturated fatty acids and you have your unsaturated fatty acids. And it's circled in red here. There's that carbon double bonded to an oxygen. So that's where those words acid are coming from. If it has a C double bonded to an O, it's called a carboxyl acid group. And that's why they have those names. And let's see, it's got one more, and I'm drawing a blank what other answer that could be. It could be lipid would be the right answer. Protein would have a C double bonded to an O. So any of those would be good answers for number 16. It's asking you for 4. There's probably more than 4 that are possible answers. All right, number 17, the ones that have nitrogen. So really importantly, if you look at the fatty acids, no nitrogen. The only thing they have, lots of carbon, lots of hydrogen, and 1 or 2 oxygens. That's all they have. If you look at the carbohydrates, that's why they're called carbohydrates. It's basically carbon, oxygen, and hydrogen, nothing else. So the ones that are left would be proteins. And again, proteins are made of amino acids. So amino acids have nitrogen. Proteins have nitrogen. And then the other one that has nitrogen are our nucleic acids. That's the DNA and RNA. Let's see, I was trying to find one where you could see it. Nitrogenous base right here. So do you see that there's nitrogens? That would be, the nitrogenous base is the A, T, C, G, or U. Do you remember A across from T and C across from G? That's that part of DNA that we match up, the A and T and C and G. All right, hopefully that answered. And if I'm going too fast, shout out, ask a question. Number 18, the sugars, those are all the carbohydrates. So you could answer lots of things. You could say monosaccharide, you could say disaccharide, you could say glucose, or you could say carbohydrate. So all of those would be examples of sugars. I only gave you 3 blanks, but I said 4. So any of those would be correct answers. Cellulose. Ooh, that's a tricky one. We haven't talked about that one yet. Cellulose is right here on the carbohydrate presentation. Cellulose is a carbohydrate. It has many, you can see it right here. It has many rings put together. So that makes it a polysaccharide. And I guess that's all. Is it just, yeah, those two. So a carbohydrate and a polysaccharide. Cellulose is what plants make. And it's why you can't eat most plants. So like leaves are made of cellulose, and it's got tons and tons and tons of sugar in it, but you don't have an enzyme to break that sugar up, so you're not able to digest it. When your dog, if you have a dog, eats grass, sometimes it throws it up, and that's because your dog doesn't have the enzyme for digesting cellulose either. All right, primary part of a cell membrane. That, dears, is something that you learned a long time ago, but have probably forgotten. Those are the phospholipids. Sometimes people draw them like this. It's a circle with 2 legs, and it looks like a little stick figure that like a kindergartner or maybe younger would try to draw. So that's a phospholipid. Yep. And so number 21, the head of the phospholipid, it has 2 words associated with it. The head is hydrophilic, which means it loves to be close to water. And the head is polar. Water is a polar molecule. It has a little bit of a charge. The head of the phospholipid has a little bit of a charge. And so they're attracted to each other, kind of like magnets are attracted to each other. Whereas now we get to number what are we on? Number 22. The tail is exactly the opposite. The tail of the phospholipid, these little blue legs, they are hydrophobic because they are nonpolar. That means they get repelled from water because they don't have a charge like water does. So those tails turn in and the heads aim toward the water. And it's not any magic. It just, if you threw a whole bunch of phospholipids in, the tails would turn away from the water and the heads would line up against the water or with the water. All right, number 23, what's a solid fat? Do you remember I do dumb things like that? Help you remember words with alliteration. A solid fat at room temperature is a saturated fat. So saturated fats have single bonds. If you look here, all the bonds between the carbons are single. The only exception is this one double bond over there, and that one doesn't count. That's the carboxyl acid group, so we're not talking about that. But all the other bonds are single, so that's a saturated, and they're solid at room temperature. The liquid fats, the unsaturated fats, have at least one double bond. So that's question number 24. That would be an unsaturated fat, would be a liquid fat at room temperature. All right, 25 kind of answered this, only single bonds in the hydrocarbon chain. So that would be single bonds is saturated. Some double bonds in the hydrocarbon chain, I'm on 26, would be the unsaturated fat. All right, number 27, we kind of looked at this already. Humans use 20 of these, so that would be amino acids. We put 20 different amino acids together to build all of our proteins in the same way that we put letters of the alphabet together to build all the different words of the English language. Number 28, the formation of a larger molecule, give me a second, I'm going to show you an image of that, is called a dehydration synthesis reaction. We've seen that a couple times this unit. When you put two molecules together and you remove water, notice the water is highlighted in this diagram. When you pull that water out and then the molecules get glued together, that's called dehydration synthesis. So that's making a larger molecule. But the same reaction can go the opposite direction if you add water to it. So as soon as you put these molecules into water, they break up into their monosaccharide form. So into their single rings instead of their double rings. And then finally, number 30, found in DNA or RNA, is it nucleic acid? Yeah, so nucleic acid, letter L, is what we're looking for there. Okay, that was the hardest section, you guys. So now I'm going to move on to, I'm going to actually show you on my computer how to draw these. If I can, oh, it's on page 2, isn't it? Get this big. How's everybody doing? Does anybody have any quick questions or anything while I'm pulling this up? Okay, so this says draw the approximate shape of a mono single sugar, di double sugar, and a 3 polysaccharide. So all you need to do for these diagrams is just draw hexagons. So I'm going to fold this over, fold my computer over, and you won't be able to see my face for a few minutes, but here we go. You're going to face up at the ceiling. But if we're going to draw a monosaccharide, the monosaccharide looks like this. Now, I could get more detail. I could put oxygen in the corner, and I could do all kinds of other things, but that is a single sugar, so that's my mono. Then I could show a disaccharide exactly the same. You put kind of like a little V in between them. And this is your disaccharide. It's got 2 hexagons put together. And then a polysaccharide would be 3 of them put together. And I am not very good at drawing hexagons. Is anybody else having trouble drawing hexagons? And that would be an example of a polysaccharide. An example, so this, I'm going to give you a minute to draw, and I'll just yammer. This could be glucose. That would be an example. You don't need to write that down. This could be sucrose, which is table sugar. So these two together would be sucrose. And this could be starch or cellulose. Starch would be something that you'd get in spaghetti or something like that. All right, next page, we'll draw the next ones. It says draw a saturated fatty acid and an unsaturated fatty acid. So I'm going to switch colors here really quick. So the fatty acids all have those really long hydrocarbon chains. So you're going to have just carbon after carbon after carbon. I don't know, put a half dozen, who cares? It doesn't matter how many of them. At one end, you have that carboxyl acid group, and it looks like this. I'm not that concerned that you know that, but just this is a saturated fat because it's all single bonds. So I'm going to call that a sat fat. Now, if you notice, if you remember HONC, pull the camera back up. If you remember HONC, hydrogen, oxygen, nitrogen, carbon, carbon is supposed to have 4 bonds around it. So that means that each of these carbons only has 2. I need to be adding all the hydrogens. So I'm going to go ahead and add those hydrogens right now. And yeah, that gets a little tedious. So, you know, if you skip them, I don't care, as long as you know that they're there. But now I'm showing the carbons. When I'm done, they will all have 4 bonds, and each hydrogen will have 1 bond, and the 2 oxygens have 2 bonds. And so they have followed the HONC rule. All right, so that's a saturated fat. And then an unsaturated fat is exactly the same, only some point along the way it has a double bond. So I threw a double bond in there. I'm going to put the carboxyl acid group at one end again. So there's my carboxyl acid group. And I'm going to add my hydrogens, but watch carefully for the hydrogens. Notice that these 2 carbons right here, they already have 1, 2, 3 bonds each, so they only get to have 1 hydrogen apiece. So I'm going to put a hydrogen here, and I'm going to put a hydrogen here, but I'm not going to put a hydrogen on the other side of them. So there's no hydrogen here, and there's no hydrogen here. Those have to go away. Okay, whoops, draw that and keep going with the same color, whatever color that was. So this is an example of an unsaturated fat. And can anyone tell me what happens to an unsaturated fat? What does the shape do? I did not indicate that in my diagram, but because there's this open space, what does that do to an unsaturated fat? Anybody?
It bends.
Nice. So an unsaturated fat has a little bend to it. And that's actually super important to your heart and to your blood vessels. That bend actually causes them to not clog, which is a funny thing. Don't get into the details. But the saturated fats pile on top of each other, and then they clog your arteries. The unsaturated fats don't make those clogs. And so those are healthier fats. So butter is saturated fat, whereas olive oil is unsaturated fat. So generally they're saying stay away. If you're somebody that has problems with that, stay away from butter and cook with olive oil. I try to cook with olive oil all the time, or other vegetable oils. All right, drawing number 33. Draw a phospholipid and label the hydrophilic and hydrophobic parts. So that's really the simplest of the drawings. We don't need to draw anything that'll take any time. That's the one that looks like a person. This is the head, and the head is hydro-loving, so it is hydrophilic, or we could say it's polar, it's both. And then the tails, are hydrophobic. They don't like water because, oops, I'm having trouble writing and talking, because they are nonpolar, so they are repulsed by water, and so they are hydrophobic. They push away. All right, number 34 is crazy hard to draw. I would never expect you to draw this on your own. I would expect you to recognize it on the test. So I would give you a picture of it and you would recognize it. I would never ask you to do this on your own without having notes available and everything like that. So you can just kind of copy what I'm doing right now. So let's choose green. So the first thing is that we have an amine group on one end of an amino acid. Give me a second. I'm wondering what all I needed to label. Okay, so then we have carbon, and we have the carboxyl acid group on this end. Do you recognize the carboxyl acid group? That was the same. This group was also present in our fatty acids that we had on question number 32. So this we can label it. This is the carboxyl acid group. And then, let's see, I'll change colors, whatever. And this is our amine group. And it's where we get the name amino acid, is from the amine group. And then carbon still has just one little hydrogen that's on the top. It doesn't have any fancy name. But then the really important thing on the bottom is the R group. And you really need to remember the R group. The R group is the part of every amino acid that's different. So we have 20 different amino acids, and they have 20 different R groups. All right, number 35, draw a dipeptide. So this is a mini protein made of just 2 amino acids. So basically it's that drawing twice, but then it's going to have a bond between them, and that's a dehydration synthesis reaction that causes them to bond together. Again, I'll draw it here quickly. I would never ask you to draw this yourself without notes. This is something that I would expect you to recognize on the test, but never to have to draw on your own. Once you take biochemistry in college, then you would have to draw this on your own. So right here, this is the bond between, this is called a peptide bond. And that's the bond between the 2 amino acids. So here's my first R group, and then this is double bonded to an O, and then now that carbon has 4 bonds, and now this nitrogen has 1 hydrogen. Now it has 3 bonds, and then we finish off with a carboxyl acid group, a little hydrogen, and an R group. And now what I usually do just to check my work is I make sure that everybody has the right number of bonds. So I see, for example, with this nitrogen, I see it has 3 bonds, 1, 2, 3. With this carbon right here, I see it has 1, 2, 3, 4 bonds. So I'm just looking to make sure that I drew it correctly, and it feels like everybody has the right number of bonds. So that would be a baby protein that we just drew. All right, let's wrap up this page with enzymes. Enzymes was the last set of notes. I haven't pulled those up recently. Actually, maybe I'll do that right now because you probably don't need me to draw. So I'll pull those notes up. So the question says, let me pull it up on my other computer. It says blank speed reaction. So let's try this page right there. Blank starts with a C, speeds reactions without being changed themselves. So those are catalysts, and catalysts can be non-biological or biological. And if it's a biological catalyst, if it's something that's made by a body or a plant, then we call it an enzyme. It's a type of protein in that case. So enzymes are a type of organic. Organic means it's biological. So it's a type of organic catalyst made of, and I just said it, made of protein. And enzymes work by fitting like bendy puzzle pieces. I'm going to show you that one where we were right there. So the gray blob in this diagram, that's the enzyme. And then you could imagine, like when we did our liver lab, the light green molecule where it says substrate, that could be the hydrogen peroxide. It touches the enzyme, and then the enzyme breaks it apart into 2 different products. And in the liver lab, those products were water and oxygen bubbles. So that's an example of what an enzyme looks like. So then finally, number 37 says, explain what the graph to the right is showing. And that is wickedly complicated for biology students. You will study this again in chemistry class. But basically, if we wanted hydrogen peroxide to bubble all by itself, we would have to add a lot of energy, and that's called activation energy. I could get hydrogen peroxide to bubble if I shook it really hard. That's adding energy. I could get it to bubble if I boiled it. That's adding energy. I could get it to bubble if I put it outside under sunlight. Ultraviolet radiation could get it to bubble. Or I could drop an enzyme or a catalyst in it, and I could get it to bubble super fast without adding a lot of energy. So it's that graph is showing that you need less energy to get the reaction to happen if you have a catalyst present. So explain what the graph to the right is showing. The activation energy is lower with an enzyme. That would be a super fast way of saying it. The activation energy is lower with an enzyme. All right, let's see if we can move on to the next page. So it says in the lab, this was from our biochemistry lab. I'm so sorry that you guys didn't get to do the labs because these labs are like mad scientist fun labs. They change colors, everybody's doing stuff. They're cool labs. Anyway, who remembers? Can anybody remember what the Benedict's test? It turned kind of lots of different colors. It turned yellow, orange, red. Does anybody remember what the Benedict's test tested for? You didn't get to do it yourselves, so you're less likely to remember, but anybody? Give you a second. Wait time. Okay, the Benedict's test is the one that, it was one of the blue tests, and it tested for sugars. Specifically, it tests for mono and certain disaccharides. The Buret's test is what tested for protein, and it turns sort of a light purple. And then the iodine tests for a type of carbohydrate, but it's specifically the specific carbohydrate it tests for is starch. So it says, oh, and then what is not soluble in water? Okay, I have a reason. I want you to pay attention to this section, so give me a second. So fats are not soluble in water. So lipids is the fancy word. Lipids are not soluble in water. And then blank and blank both have nitrogen, and we did that a little bit earlier, but proteins have nitrogen and DNA, RNA. So those are nucleic acids. They both have nitrogen. So on the test, now I said this is a formative test unless you want me to make you a summative test, and I can do that, but this is formative. I'm going to give you some test tubes, and I'm going to say how they tested. I'm going to say this test tube turned red in Benedict's, but the iodine stayed the same color, the Buret stayed the same color, and it's blah, blah, blah. And you have to be able to tell me what kind of a molecule was in that test tube. So if it turned red in the Benedict's, if the Benedict's changed color, then you would know that's a mono or disaccharide. If it changed color in iodine, you would say, oh, KJ, that's a starch or something. So just that's how you figure it out. It's one you're not going to memorize. It's one you're going to solve when you get there. Okay, off to the liver lab. So we did the liver lab, and we had some graphs that we had to do. And I think maybe I will do this one back on this page here so that you guys can see my graphs, make it really big. So it says in the liver lab, we looked at several factors that affect enzyme reaction rates. Fill in the lines for the following graphs. So the effective temperature on the enzyme reaction rate. So this is what it was supposed to be, but it didn't necessarily turn out that way. So whatever, but let's just do it the way it was supposed to be. For temperature, the enzymes work best at your body or room temperature. That's their peak. So that's where they would be up here. But they don't do well, if you remember, when we boiled them. So if you have a super high fever over 108 degrees, your enzymes, I'm going to say they melt. The right word is they, oh my gosh, the word just slipped right out of my mind. Enzymes. I don't know. The word denature. Thank you. Why did that word slip out of my mind? Thank you very much. They denature at high temperatures over 108 degrees, so they don't function at all. So that would be down there. And then cold enzymes, if your temperature is cold, they don't get damaged. Those enzymes are perfect and they're ready to function, but they're moving really, really slow. So the reaction rate is slow. So what you should have, if I'm going to make it as a line graph, I would see a line graph that went like this. That would be a good line graph. It's actually kind of a crooked line graph, but that shows you how enzymes react. They're slower when they're cold. They speed up when they're warm. And then when the temperature gets too high, they're ruined. They never work correctly. pH is similar. If that enzyme lives in your blood, then it wants a neutral pH, and we'll say neutral pH is right in the middle. So if that enzyme lives in your blood, that's where it's going to do best, is if the pH is neutral. But if you switch that pH to be acidic or basic, then it's not going to work as well. So you'll get a graph that looks a little bit like that. If the enzyme lived in your stomach, I'm just going to draw it for the fun of it. If the enzyme lived in your stomach, it's going to want a pH that's lower. So its graph is going to look more like this, and it'll peak at a lower pH. And if, I'll do the other one just for fun, because I have a demented sense of fun, if your enzyme lived in your intestines, the intestines have a really high pH, so their peak would be over there. Do you need to know all that? No, we'll just go with this. If you have the general idea of, that some pHs are good and some pHs are bad, you're good to go. And then the last one, enzyme concentration on reaction rate. So when we squished up the liver, the whole liver and the squished up liver, they both scored a 10. But this is really about how fast it reacts. So the whole liver, I'm going to write the word whole, the whole liver versus the crushed liver. We could also say, maybe what would have been better if I said low concentration and high concentration of enzymes. If it's a low concentration of enzymes, it's going to have a lower reaction rate. And if there's lots of enzymes, it's going to have a higher reaction rate. So in that case, it would look something like that. You would see a line going up to a certain point. Actually, for those of you that are interested, there's a point where it can't go any faster, and then it levels off. All right. Friends, we are almost done, and it's just about exactly what I was hoping for in terms of timing. So pulling it all together, if we had to pull everything that we've learned together, it says body temperatures above 108 degrees can cause proteins to, and thank you for helping me, to denature, which means to melt. Enzymes are proteins, so high temperatures can cause enzymes to change shape. Since enzymes work by fitting like puzzle pieces to their substrates, a changed shape means the enzyme can no longer function. Likewise, a mutation in DNA can change the shape and therefore the functioning of a protein. So some of us are lactose intolerant. Lactose is a disaccharide. It's got two sugars put together. And some of us, I'm going to go back to the, well, I can't. Some of us can't digest lactose. And that's because we have a mutation that makes our enzyme the wrong shape for pulling lactose apart. So if our DNA is different, it can change our enzymes. And then lastly, a mutation is a change in the nucleic acids. We learned about nucleic acids. DNA is deoxyribonucleic acid. This leads to a change in the order of the amino acids of the protein. Remember that all DNA does is tells our body how to build proteins. It tells us which amino acid to put after which amino acid. So, which in turn changes the shape and the functioning of the protein. Oh my goodness, peeps, we are done. Does anybody have any questions? Hey, Sam Sorensen, I didn't see you there. I don't think I said hello to you.
Hello.
How's it going?
Good. How are you?
I am good, thanks. Everybody, we are done. Does anybody have any questions or does anybody need anything from me? That was pretty fast and pretty intense, but I feel like we got through it. Did anybody?
Ms. Johnson?
Yes.
On the last assignment, the game, there's a thing that's called triglyceride.
A triglyceride?
Yeah. Can you explain what that is?
Okay. And say that again.
Can you explain what that is?
Yeah, so a triglyceride is here. Triglycerides, can you see it okay? I'm not presenting. Hold on. I forgot. I stopped presenting. Give me a second here. Oh my gosh, I'm screwing everything up. Hold on. Such a good question. Present now your entire screen. There we go. Triglycerides are under the lipids presentation. It's almost presenting. Give me just a moment. And a triglyceride is on this page. Can you see the triglyceride?
Yes.
Okay, so let's make that go away. So a triglyceride, I wish I could draw. I can't draw on this particular page, but on the far left is just what's called glycerol, and you don't need to know that. But if you look, there are 3 hydrocarbon chains going across. Those are 3 fats. They're 3 lipids. So that's where the name tri comes from. Tri is the number, there's a prefix for 3. So there are 3 fats that are going across, and those fats can be saturated fats or they can be unsaturated fats. But this gigantic molecule is how your fat cells store your extra energy. So if you eat too much and you don't burn it off during the day, then it builds these giant storage molecules that are called triglycerides. How about that? Did that help?
Yep.
Okay.
Thanks again.
Yes. Anybody else? I see over here, Ayub, you said, can you repeat the last two? And I wasn't looking, so I didn't see. Did you? I don't know if you got that, what you needed. I hope that you did.
Can you do the one with the 108 degrees Fahrenheit one?
Yes. So give me just a second. Pull it up. I gotta be in the right place. 108 degrees. Is that where this one here?
Yeah, that one.
Okay, so yeah, okay, I see it. So body temperatures above 108 degrees can cause proteins to denature, which means they melt, they fall apart. So when we have a really, really high fever, maybe you know somebody that had a really high fever, it's more common in children than it is in adults. But when they have that super high fever, it can cause brain damage, it can cause blindness, deafness, and death. There was a very famous Vikings player, Corey Stringer, who died right when you guys were little kids. And he was practicing with the Vikings on a very hot day, and his body temperature got to something like 112 degrees. I can't remember. But he died of total organ system failure, and that's because all of the enzymes in his body, all of the proteins in his body started to denature, and there's nothing doctors can do to fix that. Enzymes are proteins, so the high temps cause the enzymes to change shape. Moving on, a mutation is a change in the nucleic acids. This in turn, or this leads to a change in the order of amino acids of the protein, which in turn changes the shape and functioning of the protein. Good? Anything else, my sweets? Okay, you know how to contact me. You guys are awesome about contacting me, so let me know. I will just mention one last thing, that this is our last week of new curriculum, but then the following week, you still have to do attendance check-ins. The state is requiring that we still have school that last week. It's just that it's a makeup week, so lots of you don't have anything to do for me, but you still have to check in with all of your classes. So just make sure that you do your daily check-ins. So the following week, you do your daily check-ins. But this week is the last week of new stuff. I hope that you have a fabulous day. Yeah, I'm going to go ahead and stop the recording then. Bye, everybody.
Okay. Bye, Ms. Johnson.
Thanks. See ya, Tan.
Thanks. Bye.
Bye, KJ.
Who just said bye? I don't know who that was. Was that Ayub again?